Here's a detailed paragraph-style lecture on the difference between parametric and non-parametric statistics, tailored for CSIR Net Life Sciences, gate ecology and evolution, and other competitive exams. This explanation is structured to help in both comprehension and exam writing. Parametric versus non-parametric statistics, detailed lecture. Parametric and non-parametric statistics are two broad branches of statistical inference used for analyzing data, each with its own assumptions, applications, and strengths. The fundamental difference between the two lies in their assumptions about the population distribution from which a sample is drawn. Parametric statistics rely on the assumption that the data follow a specific distribution, most commonly the normal distribution. These methods require knowledge of certain population parameters, such as the mean, mu, and standard deviation, sigma, and are used when the underlying population is assumed to be continuous and well-defined. Examples of commonly used parametric tests include the t-test, z-test, ANOVA analysis of variance, and linear regression. These tests are powerful and efficient when their assumptions are met, meaning they have a higher probability of detecting a true effect statistical power. However, if the assumptions, especially normality and homogeneity of variances, are violated, the results of parametric tests may be misleading or invalid. In contrast, non-parametric statistics do not require the assumption of any specific population distribution. These tests are also known as distribution-free tests, and they are especially useful when the data are ordinal, ranked, skewed, or not normally distributed, or when sample sizes are small. Non-parametric methods are more flexible and robust in these situations, as they are based on medians, ranks, or signs, rather than means and standard deviations. Common non-parametric tests include the Mann-Whitney U test, alternative to independent samples t-test, Wilcoxon signed rank test, alternative to pair t-test, Kruskal-Wallis test, alternative to one-way ANOVA, and the chi-square test used for categorical data. While non-parametric tests are less powerful than parametric tests under ideal conditions, they are more reliable when assumptions are violated. In biological research, both approaches are important. For instance, if one is comparing the body weight of two animal populations with a large sample size and data are normally distributed, a parametric t-test is suitable. But if the data are non-normal, contain outliers, or are measured on an ordinal scale, e.g., pain score severity rank, a non-parametric Mann-Whitney U test would be preferred. In ecological field studies, where data are often skewed due to environmental variability or small sample sizes, non-parametric tests provide a safer statistical framework. Another distinction is that parametric methods allow for estimation and confidence intervals of population parameters, while non-parametric methods usually do not yield such estimates directly, though modern computational techniques like bootstrapping can be applied. Moreover, non-parametric methods are often easier to compute manually, which historically made them popular before the rise of statistical software. Ultimately, the choice between parametric and non-parametric methods depends on the type of data, sample size, research question, and whether the assumptions of parametric tests are met. It is generally advisable to test for normality, e.g., using the Shapiro-Wilk test or QQ-plot before applying parametric methods. If normality is not supported or if the data are ordinal or nominal, a non-parametric test should be used. Previous year question, Gate Ecology and Evolution 2020Q. Which of the following is a non-parametric test used to compare two independent groups? A paired t-test. B. Wilcoxon signed rank test. C. Mann-Whitney U test. D. ANOVA. Answer: C. Mann-Whitney U test. Practice MCQs. Q1. Parametric tests are most appropriate when a data are ordinal and non-normal. B. Sample size is very small. C. Data are categorical. D. Data are continuous and normally distributed. Answer: D. Data are continuous and normally distributed. Q2. Which of the following is not a parametric test? A one-way ANOVA. B. T-test. C. Chi-square test. D. Linear regression. Answer: C. Chi-square test. Let me know if you'd like the next lecture on types of errors. Type 1. Let me know if you'd like the next lecture on types of errors. Type 1 and type 2, or statistical power and sample size estimation. You said. Confidence interval. ChatGPT said. Here's a comprehensive paragraph-style lecture on confidence intervals, written for CSIR Net Life Sciences and Gate Ecology and Evolution level preparation. This lecture covers the theory, intuition, mathematical logic, biological applications, and past exam relevance. Confidence interval. Detailed lecture. A confidence interval C is a range of values derived from sample data that is likely to contain the true value of an unknown population parameter, such as the mean, proportion, or difference between means. Rather than giving a single estimate, confidence intervals provide a margin of error and reflect the degree of uncertainty or confidence associated with the sample estimate. The most commonly used confidence level is 95%, which means that if we were to take 100 different samples and compute a confidence interval from each, approximately 95 of them would contain the true population parameter. Thus, confidence intervals are a fundamental part of inferential statistics, allowing scientists to generalize sample findings to the larger population while acknowledging variability. The general form of a confidence interval for the population mean mu, when the population standard deviation sigma is known and the sample size is large and is greater than or equal to 30, is given by c equals x plus or minus c asterisk sigma square root n, where x is the sample mean, z is the z-score corresponding to the desired confidence level, e.g., 1.96 for 95%, sigma is the standard deviation of the population, and n is the sample size. When the population standard deviation is unknown and the sample size is small, n and lt, 30, we use the t distribution instead of the z distribution, and the formula becomes c equals x plus or minus t asterisk s square root n, where s is the sample standard deviation, and t is the t-score based on the degrees of freedom n1. In biological sciences, confidence intervals are essential in expressing precision and reliability of estimates. For example, in a clinical trial comparing two treatments, a 95% confidence interval for the difference in mean recovery times might be minus 1.2, 3.6 days. This interval suggests that while the new treatment might be faster, the range includes 0, indicating that the difference might not be statistically significant. Similarly, in ecology, confidence intervals are used when estimating biodiversity indices, population densities, or enzyme activity levels from sample data, providing a statistically defensible range rather than a fixed estimate. The width of a confidence interval depends on several factors. Sample size. Larger samples yield narrower intervals due to reduced standard error. Variability in data. Greater variability, standard deviation, results in wider intervals. Confidence level. Higher confidence, e.g., 99% leads to wider intervals, since we want to be more certain that the interval captures the true parameter. It is important to interpret confidence intervals correctly. A 95% confidence interval does not mean there is a 95% probability that the population parameter lies within the calculated interval from a single sample. Instead, it means that over many repeated samples, 95% of the constructed intervals will contain the parameter. This subtle distinction is key in avoiding misinterpretation of statistical results. Confidence intervals also help in hypothesis testing. For instance, if a 95% C for mean difference excludes 0, it suggests the difference is statistically significant at the 0.05 level. They are thus widely preferred over p-values alone, as they convey more information, both the significance and the magnitude of the effect. In modern biology and ecology, reporting confidence intervals has become standard practice in journals and experiments. It ensures transparency, reproducibility, and allows for better meta-analysis and cross-study comparisons. Previous year question, CSIR Net Deck 2014. Q. A 95% confidence interval of 1.5, 3.5 for the mean length of a fish species suggests that the population mean is 2.5 centimeters. B. 95% of fish are between 1.5 and 3.5 centimeters. C. There is a 95% probability that the true mean lies between 1.5 and 3.5 centimeters. C. Repeated sampling would yield intervals containing the true mean 95% of the time. Answer. D. Repeated sampling would yield intervals containing the true mean 95% of the time. Practice MCQs. Q1. Which of the following increases the width of a 95% confidence interval for the mean? A. Increasing sample size. B. Decreasing population standard deviation. C. Using a 99% confidence level instead. D. Increasing the sample mean. Answer. C. Using a 99% confidence level instead. Q2. A 95% confidence interval for the mean height of a plant species is 35 centimeters, 45 centimeters. Which statement is correct? A. All plants fall within this range. B. The mean of the sample is 40 centimeters. C. The population mean is definitely between 35 and 45 centimeters. D. 95% of future samples will lie in this range. Answer. B. The mean of the sample is 40 centimeters. Let me know if you'd like the next lecture on type 1 and type 2 errors, t-distribution, or p-values and hypothesis testing. You said, errors, ChatGPT said. Here's a detailed, paragraph-style lecture on type 1 and type 2 errors, essential for CSIR Net Life Sciences, gate ecology and evolution, and other competitive exams. This explanation includes conceptual clarity, real-life biological examples, and exam-focused insight. Type 1 and type 2 errors, detailed lecture. In hypothesis testing, we assess whether to reject or fail to reject a null hypothesis H based on sample data. However, since we rely on probabilistic reasoning and sample evidence, there is always a possibility of making errors. These errors are broadly classified into type 1 and type 2 errors, and understanding them is crucial for interpreting the outcomes of statistical tests in biological and ecological research. A type 1 error occurs when we reject the null hypothesis H when it is actually true. In other words, we mistakenly conclude that there is an effect or difference when none exists in the population. This is also called a false positive. The probability of committing a type 1 error is denoted by the Greek letter alpha, alpha, which represents the significance level of the test. Common values for alpha are 0.05 or 0.01, meaning there's a 5% or 1% chance of incorrectly rejecting H. In biological research, a type 1 error might occur if we conclude that a new drug is effective when in fact it is not. Minimizing type 1 errors is important in contexts where false claims can have serious consequences, such as in clinical trials or Minimizing type 1 errors is important in contexts where false claims can have serious consequences, such as in clinical trials or conservation biology. A type 2 error, on the other hand, occurs when we fail to reject the null hypothesis when it is actually false, that is, we miss detecting a real effect or difference. This is called a false negative, and the probability of making a type 2 error is denoted by beta beta. The power of a statistical test is defined as 1 beta, and it reflects the test's ability to detect a true effect when it exists. Increasing sample size, effect size, and significance level alpha can help reduce type 2 errors and increase statistical power. For example, in ecological studies, a type 2 error might occur if we fail to detect the effect of pollution on species richness even though the effect is real, possibly due to insufficient sample size or high natural variability in the data. Balancing type 1 and type 2 errors is a fundamental trade-off in hypothesis testing. Lowering alpha reduces the chance of type 1 errors but increases the risk of type 2 errors, and vice versa. The choice of alpha thus depends on the context and consequences of the error. In medical research, we may tolerate a small risk of type 1 error but prefer to minimize type 2 error to avoid missing potentially life-saving treatments. In contrast, in toxicology or conservation decisions, avoiding false positives may be prioritized to prevent unnecessary actions. Let's consider a biological example. A study tests whether a certain pesticide causes mortality in a frog population. Type 1 error, conclude the pesticide causes mortality when it actually doesn't. False alarm. Type 2 error, fail to detect pesticide-induced mortality when it actually exists. Missed risk. Each of these errors can lead to incorrect policy or conservation decisions. Understanding these errors is vital for interpreting p-values. A p-value less than alpha leads us to reject H, accepting a small probability alpha that this is a type 1 error. However, a non-significant result doesn't prove H is true. It might reflect low power or small effect size, leading to a type 2 error. Previous year question, Gate Ecology and Evolution 2021. Q. In a hypothesis test, if the null hypothesis is true but is rejected, which of the following errors has occurred? A type 1 error. B. Type 2 error. C. No error. D. Both errors. Answer. A type 1 error. Practice MCQs. Q1. Which of the following increases the risk of type 2 error? A. Increasing sample size. B. Increasing effect size. C. Decreasing alpha. Significance level. D. Increasing power. Answer. C. Decreasing alpha. Significance level. Q2. The probability of committing a type 1 error is represented by a beta. B. 1 alpha. C. Alpha. D. 1 beta. Answer. C. Alpha. Let me know if you'd like the next lecture on statistical power, p-values and hypothesis testing, or t-distribution and degrees of freedom. You said. Levels of significance. ChatGPT said. Here's a detailed, paragraph-style lecture on levels of significance, written in an exam-oriented manner suitable for CSIR Net Life Sciences and Gate Ecology and Evolution. This lecture provides the theoretical core, mathematical interpretation, biological relevance, and exam application. Levels of significance. Detailed lecture. The level of significance, commonly denoted by the Greek letter alpha, alpha, is a critical concept in hypothesis testing. It represents the probability of rejecting the null hypothesis H when it is actually true, i.e., the risk of making a type 1 error. In simpler terms, it defines how much uncertainty or tolerance for error researcher is willing to accept when drawing conclusions from statistical data. The level of significance is set before performing a test and guides the interpretation of the p-value obtained from the analysis. The most commonly used levels of significance in scientific research are 0.05, 5%, 0.01, 1%, and 0.10, 10%. An alpha of 0.05, for example, means that there is a 5% chance of falsely rejecting a true null hypothesis, a false positive result. If the p-value obtained from a statistical test is less than or equal to alpha, we reject the null hypothesis, considering the result statistically significant. If the p-value is greater than alpha, we fail to reject the null, suggesting that any observed difference could be due to chance. The choice of significance level depends on the context of the study and consequences of error. In medical or toxicological studies where a type 1 error could lead to harmful consequences, e.g., approving an unsafe drug, researchers may use a stricter alpha like 0.01. In ecological fieldwork or exploratory biological studies where a more lenient threshold is acceptable, 0.05 or even 0.10 may be used. It's important to note that reducing alpha decreases the chance of type 1 error but increases the chance of type 2 error beta potentially missing a true effect. Thus, a balance between alpha and beta must be achieved based on the scientific objective. Levels of significance are also directly related to confidence levels. For example, a significance level of 0.05 corresponds to a 95% confidence level, meaning we are 95% confident that our statistical conclusion is correct. In the case of a two-tailed test, alpha is divided into two equal parts, with each tail of the distribution having alpha 2. This is done when we test for any significant difference, regardless of direction. In biological sciences, significance levels are applied in evaluating experimental results, such as comparing mean growth rates, enzyme activities, population densities, or survival rates between control and treatment groups. For example, in a study testing whether a nutrient supplement increases plant biomass, a p-value of 0.03 would be considered statistically significant at alpha equals 0.05, and we would conclude that the supplement has a real effect on growth. It is important to interpret the significance level with caution. A statistically significant result does not always imply biological or practical importance. Conversely, a non-significant result does not always mean no effect exists. It might result from small sample sizes or high variability. Hence, effect size, confidence intervals, and replication should be considered alongside alpha for robust conclusions. Previous year question CSIR Net Deck 2018 Q. A researcher sets a level of significance at 0.01. What does this imply? There is a 1% chance of accepting a false hypothesis. B. There is a 1% chance of making a type 1 error. C. There is a 1% chance of making a type 2 error. D. The confidence level is 99% answer. B. There is a previous year question CSIR Net Deck 2018 Q. A researcher sets a level of significance at 0.01. What does this imply? There is a 1% chance of accepting a false hypothesis. B. There is a 1% chance of making a type 1 error. C. There is a 1% chance of making a type 2 error. D. The confidence level is 99% answer. B. There is a 1% chance of making a type 1 error. Practice MCQs. Q1. A test is conducted at the 5% level of significance. Which of the following is true? The test will not make any error. B. The chance of making a type 1 error is 0.05. C. The chance of making a type 2 error is 0.05. D. The p-value must be exactly 0.05 answer. B. The chance of making a type 1 error is 0.05. Q2. What happens if we decrease the level of significance from 0.05 to 0.01? We become more likely to detect a true effect. B. Type 2 error increases. C. Type 1 error increases. D. Confidence level decreases. Answer. B. Type 2 error increases. Let me know if you want the next lecture on p-value and hypothesis testing or any other topic from your statistics syllabus. You said, regression ChatGPT said. Here's a detailed paragraph-style lecture on regression analysis, designed especially for CSIR Net Life Sciences, Gate Ecology and Evolution, and other postgraduate level competitive exams. The explanation includes theoretical depth, application in biological sciences, and related practice questions with answers. Regression analysis, detailed lecture. Regression analysis is a statistical method used to examine the relationship between a dependent variable and one or more independent variables. It helps predict the value of a dependent variable based on the known values of the independent variables, and is widely used in biology, ecology, and life sciences to analyze experimental and observational data. The most commonly used form is simple linear regression, which models the relationship between two continuous variables using a straight line. When more than one predictor is involved, it is called multiple regression. In simple linear regression, we study the relationship between a single independent variable x and a dependent variable y by fitting a regression line of the form y equals a plus bx plus epsilon. Where y is the dependent variable, x is the independent variable, a is the intercept value of y when x equals 0, b is the slope or regression coefficient indicating the rate of change in y per unit change in x, and epsilon is the random error term. The slope b is calculated in such a way that the sum of the squared differences between the observed and predicted y values, i.e., residuals, is minimized. This method is called the least squares method. The regression coefficient b indicates the direction and strength of the relationship. A positive slope indicates a direct relationship, y increases with x, while a negative slope indicates an inverse relationship, y decreases with x. If b equals 0, there is no linear relationship. In biological studies, regression is useful for predicting values and understanding causal or correlational relationships. For example, researchers might use regression to predict body mass from bone length invertebrates, or to assess how temperature affects enzyme activity. In ecological studies, regression can help model how species richness changes with altitude or pollution levels, or how primary productivity is influenced by rainfall. Regression analysis also provides important metrics such as the coefficient of determination, r squared, which represents the proportion of variance in the dependent variable explained by the model. An r squared of 0.85 means that 85% of the variation in y is explained by x. However, a high r squared does not imply causation, it only suggests a strong association. It's important to distinguish correlation from regression. Correlation measures association and direction between two variables, without distinguishing between dependent and independent variables. Regression, on the other hand, predicts or explains how one variable affects another, with a clear distinction between predictor x and response y. Regression assumes certain conditions: 1. Linear relationship between variables; 2. Homoscedasticity, constant variance of errors; 3. Independence of observations; and 4. Normally distributed residuals. If these assumptions are violated, the regression model may produce biased or unreliable results. In such cases, nonlinear regression or non-parametric methods may be used. In multiple regression, we extend the model to include multiple predictors: y equals a plus bx plus bx plus bx plus epsilon. This is useful in complex biological systems where multiple factors jointly influence a response variable. For example, predicting plant growth based on sunlight, water, and nutrient availability. Previous year question: Gate Ecology and Evolution 2020. Q. In a simple linear regression analysis, the regression coefficient slope was found to be positive. What does this indicate? The dependent variable increases with the independent variable b. The dependent variable decreases with the independent variable c. No relationship exists between the variables d. The variance of y is negative. Answer. The dependent variable increases with the independent variable. Practice MCQs. Q1. The coefficient of determination r squared in a regression model indicates the correlation between two variables b. The significance level of the model c. The proportion of variance in y explained by x. D. The slope of the regression line. Answer. C. The proportion of variance in y explained by x. Q2. If a regression model has a slope of 0, what does it mean? A. X and y are perfectly correlated. B. Y decreases with x. C. Y does not change with x. D. The model is nonlinear. Answer. C. Y does not change with x. Let me know if you want the next lecture on correlation analysis, multiple regression, or ANOVA analysis of variance. Here's a detailed paragraph-style lecture on correlation, tailored for CSIR Net Life Sciences, Gate Ecology and Evolution, and similar postgrad level exams. This lecture includes core theory, interpretation, biological examples, and practice questions with answers. Correlation, detailed lecture. Correlation is a statistical technique used to measure and describe the strength and direction of the relationship between two quantitative variables. It helps answer the question: do two variables move together, and if so, how strongly and in what direction? The most widely used measure of correlation is the Pearson correlation coefficient, denoted as R, which quantifies the degree of linear association between two continuous variables. The Pearson correlation coefficient R ranges from -1 to +1. A value of +1 indicates a perfect positive linear relationship, meaning that as one variable increases, the other increases proportionally. A value of -1 indicates a perfect negative linear relationship, meaning that as one variable increases, the other decreases proportionally. A value of 0 indicates no linear relationship between the variables. Values closer to plus or minus 1 signify stronger relationships, while values near 0 imply weak or no linear association. Correlation is symmetric, meaning the correlation of x with y is the same as that of y with x. Unlike regression, correlation does not imply causation and does not distinguish between dependent and independent variables. It simply assesses how two variables covary, without assuming one affects the other. In biology and ecology, correlation is often used in observational studies where controlled experiments are not feasible. For example, researchers may examine the correlation between soil nitrogen levels and plant species richness, or between body length and metabolic rate in lizards. These relationships help formulate hypotheses but require further testing for causality. Spearman's rank correlation coefficient, rho or RS, is another measure used when data are ordinal, non-parametric, or not normally distributed. It assesses monotonic relationships based on the ranks of the data rather than the raw values. This is particularly useful in biological studies involving qualitative or ranked data, such as dominance hierarchies in animals or preference rankings in behavioral assays. It is important to visualize data using scatter plots before calculating correlation to ensure the relationship is approximately linear and not driven by outliers. In the presence of nonlinear patterns, Pearson's R may be misleading even if the variables are related. Also, spurious correlations can occur due to confounding factors or coincidence, especially in large datasets. Careful interpretation is crucial. The coefficient of determination r squared derived from the square of the correlation coefficient indicates the proportion of variability in one variable that is explained by the other. For instance, if r equals 0.8, then r squared equals 0.64, meaning 64% of the variability in y is explained by x, and the remaining 36% is due to other factors. Previous year question, CSIR net June 2017. Q. Two variables x and y have a Pearson correlation coefficient r equals -0.95. What does this imply? A strong positive correlation. B. No relationship between x and y. C. A strong negative linear correlation. D. Y increases as x increases. Answer. C. A strong negative linear correlation. Practice MCQs. Q1. Which of the following statements about correlation is true? A. Correlation implies causation. B. Correlation is asymmetric. C. Correlation measures the linear association between variables. D. Pearson correlation is used for categorical data. Answer. C. Correlation measures the linear association between variables. Q2. If the correlation coefficient between two variables is 0, it means A. The variables are independent. B. The variables have a strong linear relationship. C. The variables have no linear relationship. D. There is a perfect linear relationship. Answer. C. The variables have no linear relationship. Let me know if you want to continue with ANOVA analysis of variance or go deeper into Spearman's correlation, partial correlation, or other statistical tests next. You said t-test. ChatGPT said. Here's a detailed paragraph-style lecture on the t-test, designed for CSIR Net Life Sciences, Gate Ecology and Evolution, and other postgraduate level exams. This includes essential theory, assumptions, biological examples, and exam-focused questions. T-test. Detailed lecture. The t-test is a parametric statistical test used to determine whether there is a significant difference between the means of two groups. It is especially useful when dealing with small sample sizes, typically N and LT, 30, and is widely applied in biological, ecological, and behavioral sciences to compare group responses under different experimental conditions. The test is based on the t-distribution, which is similar to the normal distribution but has heavier tails, making it more reliable for smaller datasets. There are three main types of t-tests. One sample t-test compares the sample mean to a known or hypothesized population mean. Independent unpaired two-sample t-test compares the means of two independent groups, e.g., treatment versus control. Paired t-test compares means from the same group measured twice, such as before and after treatment, repeated measures design. The general formula for AT statistic in a two-sample independent t-test is T equals XXSE, where X and X are the sample means of the two groups, and SE is the standard error of the difference between the means, which incorporates the sample variances and sizes of the two groups. To apply AT test correctly, the data must satisfy key assumptions. Normality, the data should be approximately normally distributed. Independence, observations in each group should be independent of each other. Homogeneity of variance, both groups should have roughly equal variances in the case of independent t-test. In biological sciences, the independent t-test is commonly used to compare means from two different populations. For example, one might compare the average heart rate of treated versus untreated rats, or chlorophyll content in leaves exposed to pollution versus clean air. The paired t-test is suitable for comparing measurements taken before and after a drug treatment in the same subjects, such as the growth of plants pre and post-fertilizer application. Once the t-value is calculated, it is compared to a critical value from the t-distribution table, based on the degrees of freedom tf and the desired level of significance alpha, typically 0.05. If the p-value is less than alpha, the difference between means is considered statistically significant, meaning it is unlikely to have occurred by chance. It is important to note that the t-test only tells you if the difference is statistically significant, but not if it is biologically meaningful. Thus, effect size and confidence intervals should also be considered for interpretation. When data do not meet t-test assumptions, especially normality or equal variances, non-parametric alternatives like the Mann-Whitney U test or the Wilcoxon sign rank test are used instead. Previous year question, CSIR Net December 2018. Q. A researcher compared the mean growth rates of two independent plant groups treated with two different fertilizers and found a p-value of 0.03 using AT test. What does this suggest at alpha equals 0.05? The difference is not significant. B. The two groups have equal mean. C. The difference is statistically significant. D. The test is inconclusive. Answer. C. The difference is statistically significant. Practice MCQs. Q1. Which of the following assumptions must be met for a two-sample independent t-test? A. Data should be nominal. B. Samples must be paired. C. Observations should be dependent. D. Variances of the two groups should be equal. Answer. D. Variances of the two groups should be equal. Q2. A paired t-test is most appropriate when a. Comparing the heights of male and female plants. B. Comparing weights of two different species. C. Measuring enzyme activity before and after drug treatment in the same subjects. D. Comparing leaf area in plants grown in different locations. Answer. C. Measuring enzyme activity before and after drug treatment in the same subjects. Let me know if you'd like the next lecture on chi-square test, ANOVA, or confidence intervals. You said. Analysis of variance. ChatGPT said. Here's a detailed paragraph-style lecture on ANOVA analysis of variance tailored for CSIR Net Life Sciences, Gate Ecology and Evolution, and postgraduate level competitive exams. This includes theory, biological examples, assumptions, and exam-style questions. Analysis of variance. ANOVA detailed lecture. Analysis of variance. ANOVA is a powerful parametric statistical method used to test whether there are significant differences among the means of three or more independent groups. While AT test can compare means between two groups, ANOVA extends this logic to multiple groups simultaneously, making it a crucial tool in experimental biology, ecology, and life sciences. Instead of comparing means pairwise, which inflates type 1 error, ANOVA evaluates all group means in a single statistical test based on the variance among and within groups. The basic idea behind ANOVA is to partition the total variability observed in the data into components due to between group variation and within group variation. If the between group variance is significantly larger than the within group variance, it suggests that at least one group mean is different from the others. The core statistic in ANOVA is the F ratio, calculated as F equals variance between groups, variance within groups. A high F value indicates a greater probability that the group means are significantly different. The null hypothesis H in ANOVA is that all group means are equal, while the alternative hypothesis H is that at least one group mean differs. The most common type is one-way ANOVA, which examines the effect of a single factor independent variable with multiple levels, e.g., different diets on a dependent variable, e.g., body weight. For example, a biologist may test whether different soil types affect the height of a plant species. Another example is comparing enzyme activity in response to three different pH conditions. Two-way ANOVA is used when there are two independent variables, and it tests not only their individual effects but also their interaction effect on the dependent variable. For instance, a researcher may study how both light intensity and temperature affect chlorophyll content in plants. The assumptions of ANOVA include normal distribution of the dependent variable within each group, homogeneity of variances, equal variances across groups, independence of observations. If these assumptions are violated, especially the assumption of equal variances, then non-parametric alternatives like the Kruskal-Wallis test may be more appropriate. After ANOVA indicates a significant difference, it does not specify which groups differ. To identify where the differences lie, post-hoc tests like 2P's HSD, Bonferroni, or Dunnett's tests are performed. These comparisons control for multiple testing errors. In biological sciences, ANOVA is essential when comparing multiple treatments in experiments. For example, to assess whether different concentrations of a pesticide affect insect mortality, or whether different diets influence lipid levels in rodents, ANOVA provides a structured, statistically sound method. Previous year question, CSIR Net Deck 2016. Q. A researcher studied the effect of 4 temperatures on the metabolic rate of a lizard species. Which statistical test is most appropriate to test the hypothesis that metabolic rate varies with temperature? A chi-square test. B. Paired t-test. C. One-way ANOVA. D. Regression analysis. Answer. C. One-way ANOVA. Practice MCQs. Q1. What does the F ratio in ANOVA represent? Up ratio of standard deviations. B. Ratio of within group variance to between group variance. C. Ratio of between group variance to within group variance. D. Ratio of means. Answer. C. Ratio of between group variance to within group variance. Q2. Which of the following is not an assumption of ANOVA? A. Equal variances. B. Normality. C. Homogeneity of sample size. D. Independence of observations. Answer. C. Homogeneity of sample size. Let me know if you'd like the next lecture on chi-square test, linear regression, or a detailed explanation of post hoc analysis methods. You said, X2. Test. ChatGPT said. Here's a detailed, paragraph-style lecture on the chi-square test, crafted for CSIR Net Life Sciences, Gate Ecology and Evolution, and other advanced biology-related exams. It includes core theory, assumptions, biological examples, and relevant practice questions. Chi-square test, detailed lecture. The chi-square test is a non-parametric statistical test used to determine whether there is a significant association between two categorical variables or whether observed frequencies differ significantly from expected frequencies. Unlike parametric tests like t-test and ANOVA, the chi-square test does not require assumptions of normal distribution or homogeneity of variance and is especially useful for analyzing count data or proportions. There are two primary forms of the chi-square test. Chi-square goodness of fit test determines whether a single categorical variable follows a specified distribution. For example, it can test if the number of individuals in different phenotypic classes of a genetic cross fits the Mendelian ratio, e.g., 3, 1 9, 3, 3, 1. Chi-square test of independence evaluates whether there is a statistical association between two categorical variables, often presented in a contingency table. For example, it can test if the survival of fish is independent of water salinity levels. The formula for the chi-squared statistic is chi-squared equals sigma O, E squared E, where O is the observed frequency, and E is the expected frequency under the null hypothesis. The summation is done over all categories. A large chi-squared value suggests a greater deviation between observed and expected frequencies, indicating that the null hypothesis, no association, may be rejected. The degrees of freedom df depend on the test. For goodness of fit, df equals number of categories 1. For independence in a contingency table, df equals rows 1 times columns 1. The null hypothesis H in the chi-square test always states that there is no difference between observed and expected frequencies or that variables are independent. The alternative hypothesis H is that a significant difference exists or that variables are associated. Chi-square is widely used in genetics, ecology, and behavior. For example, it can test whether offspring ratios in a cross follow expected Mendelian inheritance, or whether species distribution in different habitats is random or shows preference. Key assumptions of the chi-square test include: the data must be in the form of frequencies or counts. Categories must be mutually exclusive. The expected frequency in each cell should generally be at least 5 for the test to be valid. Biological example. In a monohybrid cross of pea plants, you obtain 75 tall and 25 dwarf plants, total equals 100. The expected Mendelian ratio is 3, 1 tall, dwarf. Expected values, 75 tall, 25 dwarf. Observed values, 75 tall, 25 dwarf. Using the chi-square formula, chi-squared equals 75 to 75 squared, 75 plus 25 to 25 squared, 25 equals 0. Since chi-squared equals 0, the observed values perfectly match expectations, and the result is not significant. Previous year question, CSIR Net, June 2015. Q. A population of flies segregates for a single gene with 2 alleles. The observed phenotype ratio is 92 red-eyed and 28 white-eyed flies. If the expected ratio is 3, 1, what statistical test should be used to verify this? A. T-test. B. Chi-square test. C. ANOVA. D. Regression. Answer. B. Chi-square test. Practice MCQs. Q1. What is the minimum expected frequency recommended in each cell for the chi-square test to be valid? Up 1. B3 C5 D10. Answer. C5. Q2. Chi-square test is used for a comparing means of two groups. B. Testing association between two categorical variables. C. Testing correlation. D. Measuring regression. Answer. B. Testing association between two categorical variables. Let me know if you'd like to continue with linear regression, or would like a comparison of parametric versus non-parametric tests next. You said, basic introduction to neutral variate statistics, etc. ChatGPT said. Here's a detailed, paragraph-style lecture on multivariate statistics with an introduction and explanation of its applications in biological sciences, tailored for CSIR Net Life Sciences, Gate Ecology and Evolution, and PhD entrance exams. Introduction to multivariate statistics, detailed lecture. Multivariate statistics refers to a set of statistical techniques used for analyzing data that involves more than one dependent or independent variable simultaneously. Unlike univariate one variable or bivariate two variable statistics, multivariate statistics are used when dealing with complex datasets where variables are interrelated and interactions or patterns among them need to be identified. In biological sciences, ecological modeling, genetics, and behavior studies, multivariate methods help uncover patterns that cannot be detected by analyzing individual variables in isolation. For example, when studying the effect of multiple environmental factors, temperature, salinity, and pH on the distribution of marine species, or analyzing gene expression data where thousands of genes are examined simultaneously, multivariate approaches become indispensable. Some of the commonly used multivariate statistical techniques include principal component analysis (PCA), a dimension reduction technique that transforms correlated variables into a smaller number of uncorrelated components, making large datasets easier to interpret. For instance, PCA can reduce hundreds of gene expression variables into a few key axes representing major variation patterns in transcriptomics. Cluster analysis groups similar objects, species, individuals, or samples based on their characteristics or distance measures. It is often used in taxonomy, microbiome profiling, and community ecology to identify natural groupings or species assemblages. Discriminant function analysis (DFA), a method used to classify observations into predefined groups based on predictor variables. It is widely used in morphometrics, where measurements from body parts are used to classify species or sexes. Multivariate analysis of variance (MANOVA), an extension of ANOVA when more than one dependent variable is tested simultaneously against one or more factors. It accounts for the intercorrelation between dependent variables, which is often seen in biological systems. Canonical correspondence analysis (CCA) and redundancy analysis (RDA), used in ecological and environmental sciences to relate species composition, response variables to environmental gradients, explanatory variables. These techniques provide insights into species-environment relationships. Multiple regression, when multiple independent variables are used to predict a single dependent variable. For example, predicting body weight based on age, diet, and activity level. Multivariate statistical analysis is particularly powerful in the age of big data in biology, such as genomics, proteomics, metabolomics, and ecological modeling. These approaches help to extract meaningful biological information from high-dimensional datasets and reveal hidden structures or interactions. Key assumptions for most multivariate techniques include multivariate normality, absence of multilinearity, and homogeneity of variance covariance matrices. Violations of these assumptions may lead to misleading results, so proper data preprocessing and transformation, like standardization or normalization, are often necessary. Biological example. In a study on fish morphology, researchers measure length, width, fin size, and gill surface area. To see if fish from different lakes form separate groups based on these measurements, they use PCA and cluster analysis. PCA reduces the four dimensions into two principal components, and cluster analysis groups the fish into natural clusters, possibly reflecting adaptation to different lake conditions. Practice MCQs. Q1. Which multivariate technique is used to reduce the dimensionality of large datasets? A chi-square test. B. Principal component analysis. C. One way ANOVA. D. Regression analysis. Answer. B. Principal component analysis. Q2. Which method is suitable for studying species environment relationships in community ecology? A. MANOVA. B. Linear regression. C. Canonical correspondence analysis. D. Paired t-test. Answer. C. Canonical correspondence analysis. Let me know if you'd like separate, detailed lectures on PCA, cluster analysis, MANOVA, or CCA next. You said. Radio labeling techniques. ChatGPT said. Here's a detailed, paragraph-style lecture on radio labeling techniques, prepared for CSIR Net Life Sciences, GATE, and PhD entrance exam preparation in zoology and biology. It includes the theory, biological applications, and exam-oriented highlights. Radio labeling techniques. Detailed lecture. Radio labeling refers to the technique of incorporating radioactive isotopes, radioisotopes, into molecules to track or quantify biological processes with high sensitivity. These isotopes serve as tracers because their radiation emissions can be detected with high precision. Radio labeling is a fundamental tool in molecular biology, biochemistry, physiology, ecology, and pharmacokinetics. It allows scientists to follow the path, localization, uptake, metabolism, and interactions of labeled molecules in living systems or in vitro setups. Radioisotopes used in radio labeling emit different types of radiation: alpha, alpha, beta, beta, or gamma, gamma rays. Among these, beta emitters such as cube H tritium to the power of 4C, carbon 14, cube squared P, phosphorus 32, and cube to the power of 5S SULFUR 35 are commonly used in biological labeling because they provide stable incorporation into biomolecules like DNA, RNA, proteins, and metabolites. The process of radio labeling involves either chemical synthesis, where radioactive atoms are incorporated during molecule formation, or enzymatic labeling, where enzymes transfer radioactive groups to specific substrates. For example, cube squared P labeled ATP can be used to track phosphorylation events in protein signaling pathways, and to the power of 4C labeled glucose can be used to trace metabolic pathways like glycolysis in the Krebs cycle. In molecular biology, radiolabeled nucleotides are used in techniques such as northern and southern blotting, DNA footprinting, in situ hybridization, and DNA sequencing, where radioactive probes allow detection of specific nucleic acid sequences without radiography. In ecology and physiology, radiolabels are used to track nutrient flow in food chains, hormone transport, or the uptake of nutrients and toxins in organisms. Radiolabeling has also been extensively used in pharmacokinetics, where drugs labeled with radioisotopes are administered to animals or humans, and their distribution, absorption, metabolism, and excretion ADME are tracked. This provides quantitative data on drug behavior over time. The detection of radiolabeled molecules is typically done using instruments such as Geiger counters, liquid scintillation counters, and autoradiography. Autoradiography involves placing radiolabeled samples on photographic film or phosphor screens, where emitted radiation creates an image corresponding to the location of the labeled molecule. Advantages of radiolabeling include extremely high sensitivity, specificity, and the ability to trace molecules in complex biological systems without disturbing their function. However, the handling of radioactive materials requires strict safety protocols due to potential health hazards from radiation exposure. Proper shielding, waste disposal, and licensing are essential in labs using radioisotopes. Short-lived isotopes, e.g., cube squared P, with a half-life of approximately 14 days, are often preferred for short-term experiments, whereas long-lived isotopes, like to the power of 4C, with a half-life of approximately 5,700 years, are used in long-term labeling studies or for carbon dating. Biological examples. Cube squared P DNA labeling to detect DNA synthesis rates or to study DNA-protein interactions. To the power of 4C labeled amino acids to measure protein synthesis. Cube H thymidine incorporation to assess cell proliferation in tissue culture. Radiolabeled hormones, e.g., squared to the power of 5 by insulin to study receptor binding and signal transduction. Practice MCQs. Q1. Which of the following radioisotopes is most commonly used to label DNA in molecular biology? A to the power of 4C. B cube squared PC cube to the power of 5SD squared to the power of 5 by answer. B cube squared P. Q2. What is the purpose of using radiolabeled compounds in metabolic studies? A to increase reaction rates. B to visualize and quantify molecular movement. C to reduce metabolic activity. D to enhance enzyme activity. Answer. B to visualize and quantify molecular movement. Let me know if you'd like the next lecture on chromatography techniques, electrophoresis, or spectroscopy methods in biological sciences. You said. Detection and measurement of different types of radioisotopes normally used in biology. ChatGPT said. Here's a detailed, paragraph-style lecture on detection and measurement of different types of radioisotopes normally used in biology, suitable for CSIR Net Life Sciences, GATE, and PhD entrance exams in zoology. Detection and measurement of radioisotopes in biology. Detailed lecture. Radioisotopes are widely used in biological sciences for tracing biochemical pathways, studying molecular interactions, and quantifying physiological processes. Their utility lies in their ability to emit detectable radiation. To harness their potential, one must understand how to detect and measure the type and intensity of radiation emitted by different radioisotopes. The choice of detection method depends on the type of radiation: alpha, beta, or gamma energy level, half-life, and the biological application. In biological research, the most commonly used radioisotopes include tritium cube H, a low energy beta emitter, used to label DNA, RNA, or proteins. Carbon 14 to the power of 4C, a beta emitter, often used in metabolic studies. Phosphorus 32 cube squared P, a high energy beta emitter, frequently used to label DNA or nucleotides. Sulfur 35 cube to the power of 5S, a beta emitter, used in protein and amino acid labeling. Iodine 125 squared to the power of 5I, a low energy gamma emitter, used for hormone and receptor binding studies. Detection instruments and techniques: Geiger-Müller GM counter. The GM counter detects ionizing radiation by producing a current pulse when radiation passes through a gas-filled tube. It is effective for detecting beta and gamma radiation, especially from isotopes like cube squared P and squared to the power of 5I. However, it lacks the ability to distinguish between different isotopes or quantify energy levels accurately. Liquid scintillation counter LSC. LSC is highly sensitive and widely used to measure low energy beta emitters, such as cube H to the power of 4C. In this method, a radiolabeled sample is mixed with a scintillation cocktail that emits light photons when beta particles interact with it. These photons are then detected and quantified by photon. These photons are then detected and quantified by photomultiplier tubes. This method is especially valuable in detecting weak beta emitters that cannot be picked up by GM counters. Gamma counter. Gamma counters use sodium iodide night scintillation detectors to measure gamma rays, especially from isotopes like squared to the power of 5I. This is essential in studies involving radiolabeled hormones or ligands, where gamma radiation penetrates tissue and can be quantified with precision. Autoradiography. This technique uses photographic film or phosphor imaging plates to detect radioisotopes. The radiolabeled biological samples are placed against the film, and the emitted radiation creates a latent image, which is developed to visualize the distribution of the isotope. Autoradiography is widely used in molecular biology, e.g., southern northern blotting, and histological localization of radioisotopes. Solid scintillation counting, dry counting. For samples on solid supports like filters or membranes, e.g., in blotting experiments, solid scintillation counters can be used. These utilize phosphor screens or detectors placed directly on the sample. Cherenkov counting. A specialized form of detection for high energy beta emitters like cube squared P, where radiation exceeds the speed of light in a medium and produces visible light. This method is useful for counting without adding scintillation fluid, though it's less sensitive than traditional LSC. Measurement parameters. Counts per minute CPM, the raw output of detected disintegrations. Disintegrations per minute DPM, the actual number of radioactive decays, calculated by correcting CPM for detector efficiency. Specific activity, radioactivity per unit mass of a compound, useful for comparing labeling efficiency. Half-life T1/2, the time required for half the atoms of a radioisotope to decay, important for experiment planning and waste management. Biological importance and safety. Detection of radioisotopes allows biologists to quantify dynamic processes such as DNA replication, protein synthesis, ligand receptor binding, and metabolic flux. However, radiation safety protocols are essential to minimize exposure. Shielding plexiglass for beta, lead for gamma, proper waste disposal, and radiation badges for monitoring exposure are standard in all radiological labs. Practice MCQs. Q1. Which of the following radioisotopes requires a liquid scintillation counter for accurate detection due to its low energy beta emissions? A cubed squared P. B squared to the power of 5 I C cubed HD to the power of 6 counting answer, C cubed H. Q2. Gamma radiation emitted by squared to the power of 5 I is best detected using a Geiger counter. B. Autoradiography C. Gamma counter with night detector D. Liquid scintillation counter answer, C. Gamma counter with night detector. Let me know if you want the next topic on RFLP, PCR, or ELISA. You said. Incorporation of radioisotopes in biological tissues and cells, ChatGPT said. Here's a detailed lecture on the incorporation of radioisotopes in biological tissues and cells, written in a paragraph style suitable for CSIR Net Life Sciences, GATE, and PhD entrance exams in zoology and biology. Incorporation of radioisotopes in biological tissues and cells, detailed lecture. The incorporation of radioisotopes into biological tissues and cells is a critical technique in experimental biology, used to trace, measure, and visualize dynamic biochemical processes with high precision. This method involves introducing radioactive atoms into biologically relevant molecules, such as nucleotides, amino acids, sugars, or hormones, which then get metabolically or structurally incorporated into the cells or tissues of interest. The labeled molecules, now radioactive, can be followed through various metabolic pathways, enabling researchers to study gene expression, cell proliferation, protein synthesis, nutrient uptake, or signal transduction events. The most common method of incorporation is metabolic labeling, where radioisotope labeled precursors are supplied to living cells or tissues in culture or to whole organisms. For example, cubed H-thymidine, tritiated thymidine, is incorporated into newly synthesized DNA during the S phase of the cell cycle and is widely used in cell proliferation studies. Similarly, cube to the power of 5s labeled methionine or cysteine can be added to the medium to label newly synthesized proteins, as sulfur is found in these amino acids. To the power of 4C labeled glucose is used to trace glycolytic flux or respiration, while cube squared P labeled phosphate can be incorporated into ATP and nucleotides, aiding in the study of phosphorylation events and nucleotide turnover. Once incorporated, these labeled biomolecules can be detected using techniques like autoradiography, liquid scintillation counting, or gamma counting, depending on the nature of the isotope. For tissue-level studies, autoradiographic techniques allow for the spatial visualization of isotope incorporation at the cellular or subcellular level using photographic film or phosphor screens. This has been extremely valuable in developmental biology and histological studies to localize gene activity or nutrient transport. In plant biology, radioisotopes like cube squared P to the power of 4C are supplied through the roots or leaves and are then tracked throughout the vascular system to study nutrient transport, photosynthetic assimilation, or hormone movement. In animal systems, isotopes can be introduced via injection, ingestion, or inhalation, and tissues can be harvested after set intervals to determine uptake and incorporation. The efficiency of incorporation depends on several factors: the metabolic activity of the cells, the concentration and specific activity of the radiolabeled compound, incubation time, and cellular uptake mechanisms. Pulse chase experiments are often used to understand the temporal dynamics of incorporation. In such experiments, cells are briefly exposed to the radiolabeled compound pulse followed by a non-radioactive version, chase, allowing researchers to study the turnover or processing of biomolecules over time. Safety considerations are crucial during incorporation experiments. For example, cube squared P, a high energy beta emitter, requires protective shielding to prevent radiation exposure, whereas low energy beta emitters like cube H or to the power of 4C pose less risk but still require proper handling and waste disposal. The radioactive materials used must be bioavailable, stable under experimental conditions, and should not disrupt normal cellular function. Examples in biology: cube squared P orthophosphate incorporation in phosphorylation studies of proteins. Cube to the power of 5s methionine for studying protein translation in cell-free systems. Cube H uridine incorporation to study RNA synthesis in cultured cells. To the power of 4C labeled sucrose to trace flow and loading and translocation in plants. Practice MCQs. Q1. Which of the following radioisotopes is commonly incorporated into DNA during cell separation assays? Examples in biology: cube squared P orthophosphate incorporation in phosphorylation studies of proteins. Cube to the power of 5s methionine for studying protein translation in cell-free systems. Cube H uridine incorporation to study RNA synthesis in cultured cells. To the power of 4C labeled sucrose to trace flow and loading and translocation in plants. Practice MCQs. Q1. Which of the following radioisotopes is commonly incorporated into DNA during cell proliferation assays? A cube squared P. B cube to the power of 5s. C cube H thymidine D squared to the power of 5I. Answer: C cube H thymidine. Q2. What is the primary method used to detect the location of incorporated radioisotopes within tissue sections? A PCR. B ELISA. C autoradiography D mass spectrometry. Answer: C autoradiography. Let me know if you'd like the next lecture on isotope dilution techniques, ELISA, or immunofluorescence methods. You said molecular imaging of radioactive material. ChatGPT said. Here's a detailed paragraph-style lecture on molecular imaging of radioactive material, tailored for CSIR Net Life Sciences, GATE, and PhD entrance exams in zoology and biology. Molecular imaging of radioactive material, detailed lecture. Molecular imaging is a powerful technique in biological and biomedical research that allows for the non-invasive visualization, characterization, and quantification of biological processes at the molecular and cellular levels in living organisms. When radioactive materials, radioisotopes, are used in molecular imaging, they serve as tracers that emit radiation detectable by sensitive instruments, allowing scientists to monitor specific biochemical events in real time within intact tissues or whole organisms. This form of imaging bridges the gap between molecular biology and in vivo physiology, enabling dynamic observation of processes such as gene expression, cell signaling, metabolism, and drug receptor interactions. Radioactive molecular imaging techniques rely on radiotracers, radioactive compounds that are designed to bind or participate in specific biological pathways. These tracers are often labeled with gamma-emitting or positron-emitting radioisotopes. For example, fluorine 18 to the power of 8F, a positron emitter, is used in fluorodeoxyglucose FDG, which is taken up by metabolically active cells and is widely used in positron emission tomography PET. Likewise, technetium 99 meters to the power of 9 to the power of 9 TC, a gamma emitter, is used in single photon emission computed tomography SPECT. Positron emission tomography PET is one of the most sensitive techniques for molecular imaging. In PET, radiotracers that emit positrons undergo annihilation reactions with electrons, producing pairs of gamma photons that travel in opposite directions. Detectors arranged in a ring around the subject capture these photons and reconstruct a three-dimensional image of tracer distribution. PET is used in neuroscience to study brain function, in oncology to image tumors, and in cardiology to assess heart metabolism. Single photon emission computed tomography SPECT is another molecular imaging modality that uses gamma-emitting isotopes. SPECT provides information about the functional state of tissues and is useful in mapping receptor density, blood flow, or metabolic activity. Unlike PET, SPECT tracers do not require annihilation reactions, and its instruments are generally more widely available and less expensive. In molecular biology, radioactive imaging is also done using autoradiography, where radioactively labeled molecules such as DNA, RNA, or proteins are separated via gel electrophoresis or blotted onto membranes and then visualized using photographic film or phosphor screens. This static imaging is vital for gene expression studies, mutation detection, and protein DNA interaction analysis. Additionally, radiolabeled antibodies or ligands are employed in targeted imaging of specific tissues or cell types. For example, antibodies labeled with square to the power of 5 to the power of 9 to the power of 9 TC can target cancer cell markers and help in the localization and diagnosis of tumors. In gene expression studies, radiolabeled nucleotide probes are used in situ hybridization to visualize mRNA expression in tissue sections. Safety and specificity are crucial when using radioactive materials for imaging. The radiotracer must exhibit high affinity, specificity, and stability, with minimal toxicity or interference in normal biological function. Proper shielding and dosimetry are required to minimize radiation exposure to both the subject and the researcher. Molecular imaging of radioactive material thus provides a dynamic, real-time view of molecular events, making it indispensable in diagnostics, drug development, functional genomics, and systems biology. Practice MCQs. Q1. Which of the following imaging techniques uses positron emitting radioisotopes for molecular imaging? SPECT. B PET CMRID Court. Answer: B PET. Q2. What is the primary radioactive tracer used in PET imaging to detect metabolic activity in cancer cells? A cube dye. B cubed H limiting C to the power of 9 to the power of 9 TCD to the power of 8 FFDG. Answer: D to the power of 8 FFDG. Let me know if you want the next lecture on isotope dilution, immunofluorescence methods, or enzyme linked assays. You said safety guidelines. ChatGPT said. Here's a detailed paragraph-style lecture on safety guidelines and the use of radioactive materials in biology, ideal for CSIR Net Life Sciences, GATE, and PhD entrance preparation in zoology and related biological sciences. Asterisk. Safety guidelines and the use of radioactive materials in biology. Detailed lecture. The use of radioactive materials in biological research demands strict adherence to safety guidelines to protect both researchers and the environment from harmful exposure. Ionizing radiation emitted by radioisotopes, such as alpha, beta, and gamma rays, can damage living cells, cause DNA mutations, and increase the risk of cancer. Therefore, comprehensive radiation safety protocols have been developed to ensure safe handling, storage, and disposal of radioactive substances in laboratory and clinical settings. One of the primary principles of radiation protection is the ALARA principle, as low as reasonably achievable, which encourages minimizing exposure by optimizing three factors: time, distance, and shielding. Exposure time should be kept as short as possible. Distance from radioactive sources should be maximized, and appropriate shielding, like lead bricks or plexiglass barriers, should be used depending on the type of radiation emitted. For example, plexiglass is commonly used to shield beta emitters like cube square P, while lead shielding is necessary for gamma emitters like cube to the power of 9 to the power of 9 TC. Only trained personnel should handle radioactive materials, and they must follow protocols set by national regulatory bodies such as the Atomic Energy Regulatory Board (AERB) in India or radiation safety committees of respective institutions. Laboratory workers must use personal protective equipment (PPE), including lab coats, gloves, and sometimes face shields. Radiation dosimeters, film badges, or TLD badges are worn to monitor cumulative radiation exposure. Laboratory areas where radioactive materials are used must be clearly marked with radioactive hazard symbols, and access must be restricted to authorized personnel. A well-ventilated, designated radioisotope work area should be used, often equipped with fume hoods or laminar flow cabinets to prevent inhalation of radioactive aerosols. Surfaces should be non-absorbent and easy to decontaminate, and spill kits must be readily available. Regular surface contamination checks using Geiger Muller counters or scintillation detectors help monitor cleanliness and safety. In case of spills, immediate containment, notification to safety officers, and proper cleanup procedures are mandatory. Storage of radioactive materials must be done in shielded, labeled, and secure containers, segregated according to isotope type and half-life. Logbooks should record the inventory, usage, and disposal details. Disposal of radioactive waste follows specific guidelines. Short-lived isotopes may be stored for decay, while others require authorized disposal facilities. Liquid waste should never be poured down the drain unless permitted, and solid radioactive waste must be incinerated or buried according to institutional and environmental regulations. Moreover, researchers must undergo regular training and refresher courses in radiation safety and emergency procedures. Emergency protocols include steps for contamination incidents, accidental exposure, and fire hazards involving radioactive materials. In institutions, the Radiation Safety Officer (RSO) is responsible for compliance with regulatory norms and training. In summary, safe use of radioactive materials is critical in biological research and demands rigorous attention to established protocols. These guidelines not only ensure researcher and environmental safety but also uphold the integrity and reproducibility of scientific research involving radioisotopes. Practice MCQs. Q1. What does the ALARA principle in radiation safety stand for? All lead and radiation approved B, as long as radiation accumulates. C, as low as reasonably achievable. D, allowable limits against radiation accidents. Answer: C, as low as reasonably achievable. Q2. Which shielding material is most appropriate for high energy beta emitters like cube square P? A paper. B, lead. C, plexiglass. D, aluminum. Answer: C, plexiglass. Let me know if you'd like the next lecture on ELISA, immunofluorescence, flow cytometry, or any other technique. You said. More about that topic. ChatGPT said. Here's a continued and expanded lecture on safety guidelines and the use of radioactive materials in biological research, providing further insight into types of hazards, biological effects, institutional responsibilities, accident management, and international standards. Extended lecture on safety guidelines and the use of radioactive materials in biology. Types of radiation hazards. Radiation hazards are generally classified into external and internal exposure. External exposure occurs when a radioactive source is outside the body, potentially affecting skin, eyes, or deeper tissues depending on the radiation type. Internal exposure, which is more dangerous, happens when radioactive materials enter the body through inhalation, ingestion, absorption through skin, or accidental injection. For example, accidental inhalation of radioactive iodine cube can localize in the thyroid and cause tissue damage. Hence, stringent protocols are followed to contain airborne isotopes and prevent internal contamination. Biological effects of radiation. Ionizing radiation interacts with biological tissues by producing free radicals, particularly through radiolysis of water, leading to DNA strand breaks, chromosomal aberrations, mutations, or cell death. Tissues with high meiotic rates, such as bone marrow or intestinal epithelium, are more vulnerable. While acute high-dose exposure may cause radiation sickness, chronic low-dose exposure increases the risk of cancer, reproductive damage, or hereditary effects. Therefore, cumulative exposure is monitored with dosimeters, and limits are set by organizations like ICRP, International Commission on Radiological Protection. Institutional responsibilities and infrastructure. Every institution using radioactive materials must maintain a radiation protection program, which includes appointment of a radiation safety officer (RSO), formation of an institutional radiation safety committee (RSC), periodic risk assessments and internal audits, establishment of controlled and supervised areas with restricted access, training programs for all radiation workers, maintenance of detailed records of radioisotope procurement, use, and disposal. The AERB in India or equivalent national bodies require that all users and institutions obtain prior authorization or licensing before procuring or using radioactive materials. Spill and accident management. In case of minor spills, e.g., a few micro-C, absorbent paper and decontamination solution are used with gloves, while contamination levels are monitored using Geiger counters or liquid scintillation devices. For major spills, e.g., involving MCI amounts, the area must be immediately evacuated, the RSO informed, and the area cordoned off. Personnel potentially exposed should undergo bioassay or medical evaluation, and all findings are documented for regulatory review. Fire or flooding involving radioactive areas requires special handling, as these can spread contamination. Fire departments must be alerted to the radiological nature of the lab, and emergency services coordinate decontamination procedures. Decontamination procedures. Personal decontamination includes removal of contaminated clothing, washing skin with mild detergent, and medical evaluation if internal exposure is suspected. Surface decontamination uses specific chemical agents based on the isotope used, such as citranox, EDTA-based cleaners, or even dilute HCl or ethanol depending on compatibility. Decontaminated materials are either labeled for decay storage or disposed of via authorized waste disposal routes. International guidelines and monitoring. International bodies like the International Atomic Energy Agency (IAEA) and ICRP issue standards for radiation protection, dose limits, and training. Dose limits for radiation workers are generally set at 20 millisieverts per year average OA.